All right, our last speaker for the session is Waterhouse from Northeastern. Just gonna wait for the signal. So our recent ability to ascertain massive amounts of sequence data has been paralleled by this rapid increase in the number of statistical analyses and statistics that we can use to analyze this data. And all these statistics measure different things, but they all have a common goal, which is to identify areas of the genome that behave differently than the rest of the genome. And these outlier areas, or these spikes in Manhattan plots, are areas that we infer might be under selection because of their behavior. But what the field has really overlooked is how to integrate or combine analyses from multiple tests to prioritize loci for further investigation. They don't have to be genes necessarily, but a common approach is to take the overlap from different statistical tests, the ones that share outliers, and those are the loci to prioritize. However, this approach has some limitations. Most notably, these statistics aren't just measuring different things. These statistics are also varying in their sensitivity to demographic history and sampling design, like my work and others have shown with simulations. And so a reasonable alternative might be to look for outliers in multivariate space. But because the results from these statistics are correlated, we can't use a simple measure of Euclidean distance. For example, the Euclidean distance from the center of this distribution to point A is much larger than it is to point B, but point B is more of an outlier in this distribution. The Mahalanobis distance was developed for this purpose, and it basically corrects for covariance among statistics in multivariate space. So the Mahalanobis distance to point B would be larger than it is to point A because of the correction for covariance, which is the S in this equation. However, Mahalanobis distance itself is limited because it assumes that data are parametric and can be described by an ellipsoid in multivariate space. So we developed three statistics that vary in their sensitivity to the density of local points so that they can be used in non-parametric situations. The first measures the harmonic mean distance between a focal point and every other point in the data set. The second is based on kernel density, which basically assumes every point in the data can be approximated by a simple parametric distribution centered on that point. And we calculate the log probability density at a focal point based on the kernel density to all other points in that data set apart from that focal point. And we also use log likelihood to estimate the bandwidth on that kernel. And finally, we developed a measure of nearest neighbor distance, and all of these measures are corrected for covariance in multivariate space by this S in the equations. So to give you an idea of these different sensitivities of these different statistics on a non-parametric distribution, the more outlier-y contours are the ones that are in yellow. And you can see that non-parametric distribution, Mahalanobis distance is approximating the data as ellipsoid. Harmonic mean distance is slightly more sensitive to the density of points in local space. Kernel density is increasingly sensitive, and nearest neighbor distance is the most sensitive to the density of local points. So we wanted to ask the question whether multivariate outliers could improve the power to detect selected loci. And we first looked at this with simulations. And these are simulations that I previously published and spoke about at this meeting. It's based on a landscape simulation of demographic expansion from a 2-refuge model. Through time is shown up here at the top. And I've combined results from multiple different sampling designs. So some of the variance that you see in the results is due to the variance in the results from these different sampling designs. And we had previously analyzed this data set with different univariate statistics. 3 of them are based on a genetic environment association. One of them is Spearman's Rho, another one is a Bayes factor, and the third one is a z-score. And we used a 4th measure of genetic differentiation that's like FST. It's called XTX. And here I'm showing you the multivariate distributions of these different loci. And in multivariate space, the neutral loci are in gray, the weak selected loci are in light blue, medium selections in dark blue, and strong selections in dark red. And these were all under selection to the same environmental gradient, which was applying on the landscape. What's interesting is I think when you start to look at these in multivariate space, the loci under weak selection are not necessarily outliers by any one univariate method, but they do start to look like outliers in multivariate space, for example, here on your bottom right. So here's the previous published results for these univariate statistics. And a lot of the variance that we're seeing in power for these different statistics is resulting from their variance in performance on those different sampling designs. We can compare that to the application of our multivariate statistics, and we don't see a very compelling pattern here. Mahalanobis distance and harmonic mean distance seem to be doing okay. Kernel density deviance and nearest neighbor distance don't seem to be doing that well. And that seems at odds with that visual plot that I just showed you. And what I wondered was whether the calculation of covariance was getting biased by the presence of selected loci in the data set. And so I compared the covariance for all data, shown here, visualized in black, to the covariance for just neutral loci, shown here in blue. And the covariance for the neutral loci was smaller, showing that the presence of the selected loci in the data set did bias the covariance. And this is just in 2 dimensions. So you can imagine that this effect adds up as you add dimensions onto the data. So the first problem is that the selected loci bias the estimate of the covariance. And the second problem was that the selected loci had correlated responses. So the loci were unlinked in the simulated data set, but because they were responding to the same selection, the nearest neighbor in multivariate space to one locus under selection was another locus under selection. So you can imagine this is even a bigger problem when you have linkage added to the equation. So what we wanted to do was figure out if we could identify a set of points in the data that was not influenced by outliers in the data set. And unlike when I developed Outplank with Mike Whitlock, we actually didn't make stuff up here. We actually consulted a large body of literature on robust statistics. And perhaps the most widely used measure in robust statistics is the minimum covariance determinant. And what this does is actually find a set of points in your data set that is not influenced by outliers. And it does that by minimizing the determinant on the covariance matrix. And geometrically, what that means is it's minimizing the volume in multivariate space that those points describe. And so when we use the MCD estimate of the covariance, in most of the simulations that I've looked at so far, it's a very good approximation of the covariance based on just the neutral loci alone. The beauty of the MCD algorithm is it also identifies which points are robust, that is, that they are not influenced by outliers. And we can use those robust points in these measures for multivariate distance. So now that we're not measuring the nearest neighbor to all points in the data set, but we're measuring the nearest neighbor to the nearest robust point. So now we can compare power based on the MCD with the robust points to just the default. And we see not only a slight increase in power, but a huge decrease in the variance of results. And so in different sampling designs, the same loci are popping up again and again as outliers in these data sets. And because the application I've done here is primarily parametric, these 4 statistics all perform very similarly. And so the one that you use will depend on the actual measurement that you want to convey in your analysis. So then we ask the question again, but this time with empirical data, and we use a recently published data set on selected sweeps in dog breeds. This was a really nice data set that was just published this year in Molecular Ecology, and it basically used loci that were known to have strong phenotypic effects and to be under selection in these different breeds, evaluated their frequency in each breed, and then compared the power of different selection scan statistics to detect selection on these loci. And so here's an example of one locus, the MC5R locus, and its relative frequency in this different breed, so highest frequency at the top, lowest frequency at the bottom. And the blue areas indicate when these statistics had very high power to detect selection. So for example, statistics like H12, Tagemas D, and nucleotide diversity had high power in cases when the allele frequency was near fixation, at or near fixation, whereas for this particular locus, IHS and NSL, the number of lengths of, never mind, I don't remember. It had more power to detect selection when the frequency of the locus was in the process of sweeping, so it was only at 0.51%. So to illustrate this, I've chosen one breed, the Havanese breed, and these Manhattan plots are for 4 of those statistics, IHS, H12, nucleotide diversity, and Tagemas D. And the vertical lines indicate the loci that we know have phenotypic effects on these dog breeds and have risen to a frequency of at least 50% in this breed. So I chose this data set because these statistics have different abilities to detect these loci, indicated here by the stars. And for example, nucleotide diversity doesn't show much of a pattern, and this 5th locus isn't really detected by any of these statistics. So we can apply one of our multivariate measures. Here I've used nearest neighbor distance with the default assumptions. And again, we don't see a very compelling pattern. But if we use the nearest neighbor distance with the MCD and the robust points are highlighted here in yellow, we do see signatures of selection at the loci that also show signatures in at least one of the univariate statistics. So for example, that 5th locus that doesn't show a signature in any of those statistics also doesn't really show a signature here. So that's another important take-home point. These multivariate summary statistics are only going to be as powerful as the univariate statistics that they're summarizing. So, these robust points, even though they were neutral in my simulations, that doesn't mean that they're neutral in the real data. Maybe the whole genome is under selection. If a point is outside that robust area, that doesn't mean that that's under selection. I'm not advocating that robust points should be used as a null model, but it might be a more objective way of identifying loci if you did want to use them for some kind of null model. What I think is also interesting is that you can go back and visualize where these robust points are for these different statistics. For example, H12, you can see at that 6th locus on the far right, really had no power to detect selection for that locus. So in conclusion, MCD and robust statistics have been underutilized for outlier detection in population genetics. These measures of covariance come up in a lot of different statistics, and there are more robust ways of estimating them that might be useful. This does seem to be a better way of prioritizing candidates across multiple tests than just taking the intersection. But you have to know your data. If your data is parametric, you can use the MCD, but because the MCD itself assumes data is parametric, if your data is not parametric, it doesn't really make sense to use it. But you could use one of the other 3 measures that we developed without the MCD. And along those lines, we are looking for some more nonlinear or non-parametric examples to test these on. So if you have data that you think would be cool, please talk to me. And finally, these multivariate summary statistics are only telling you about outliers in multivariate space. They're not really saying anything about evolution. If you want to prioritize candidates, it's a nice way to summarize your data, but they do not replace the univariate statistics that they're based on. We understand how these statistics are affected by evolutionary forces, and you need to look back at those to understand how evolution is affecting your data. So with that, these statistics are implemented in an R package called Minotaur, which is on GitHub, and I'd like to acknowledge Nessun for bringing us together. Thank you.